Good morning. This is Alana, and I work in the DNA department at the RAAA. Today, we're going to be going over reading your results. We're going to go over everything from how long does it take to see results from us, all the way to if you have an exclusion and what to do. First up, like I said, is how long does it take? So from the time you receive the DNA processing email, which you can see below on the right-hand side, if everything goes well, it takes the lab about 15 to 20 business days to process the sample and generate results. So basically, if you're kind of on a time crunch, I always say the earlier the better. There are some things that may affect that 15 to 20 business days though, such as seasonality of submissions. I always tell people the earlier the better. When we're in peak season for DNA, sometimes our length gets a little bit longer at the lab just because they're processing thousands of samples every day. Also, how many samples and what type of testing you're doing, the quality of the samples, and if they need to be reprocessed for any reason. And basically, reprocessing we'll go over a little bit later. Also, if the order form was filled out correctly. So if I'm missing any information here, I will contact you via email usually first, and then if I don't hear from you, I'll give you a call. And basically, if I'm contacting you, it's information that I need in order to submit the sample. So please make sure you watch your email or listen to your voicemail messages and make sure your mailbox isn't full. Also, we're going to go over what do you get when they're done. So there's two different kinds. Basically, we have our Red Navigator, which is our commercial female testing, or we have our Gene-Seek Parentage Defect or GGP testing. So the first one up is Red Navigator. So you will be emailed your results, usually from Fallon. And basically, these results are a percentile rank against the commercial population of Red Angus animals. So for example, a ranking of 1 would mean that that animal is in the top 1%. So if you have any questions about your Red Navigator results or comments or concerns or, what do you do now, Katie, who is on our commercial marketing team, will follow up with you via phone call. You can also reach her at extension 16 or Katie Oshner at redangus.org. Next up is our Gene-Seek Parentage Defect and GGP testing. We're going to kind of go over the different scenarios that you may see when you get your results back. So first up, basically these animals, animal ID 1, 2, 3, and 4, were submitted without registration numbers, which is okay for our parentage and defect testing. If you do, on a side note, want to submit an animal for GGP testing, it has to be registered so we can attach those results to something. So these animals were tested for parentage. Basically, you can't see that from the screen, but that's just what I know what happened. So basically, if you want these results reflected on their pedigrees, then you will need to enter in the registration number in the registration column and then send it back to Fallon. And the key here is that the member must return the file below to Fallon. So make sure you put in the registration number in the registration column, and then she can upload your results to their pedigrees. Next up, we're going to go over some kind of big ones. So we're going to go over what qualified means, excluded, and genotype not on file. So basically, qualified indicates that the parent registration number listed has been confirmed and verified through the DNA parentage test and is the parent. So you can see at that very bottom one that we have a qualified for the sire. Now going over the excluded, so just as a refresher, all sires and donor dams need to have DNA on file with RAAA. So when you have an exclusion, as you can see in that first sire column 1, basically that animal is going to go on hold because we require that they all have DNA on file with us and that we need to get the exclusion fixed so the pedigree can be reflected correctly through RedsPro. So in essence, excluded indicates that the parent registration number listed does not verify through the DNA parentage test to be the correct parentage. And it says, please review the exclusion notice letter attached for steps to resolve and additional information. So whenever you have an exclusion in your pedigree, you will get that exclusion notice. And basically, it goes over, like it says, all the steps to resolve that. And what you need to do in like this case, the very first one, is you need to give Fallon other options of who that animal could be out of as far as the sire, so we can get that pedigree corrected. So with exclusions as well, I get a lot of questions like, do I need to resubmit a new sample? And the only time that you would need to do that is if you thought that the sample was contaminated and that's why it's excluding. Now, generally, that's not the case. All you need to do is just send Fallon, like I said, other options for who the sire could be for that animal. We can run that parentage here in-house. It goes in date received order. And then once Fallon has completed it, she will send you an email saying, hey, all those other options that you gave me, I'll exclude it as well. I need some more. Or, hey, we found the parent for that animal and we're good to go. So make sure you communicate with Fallon if you have an exclusion, because your animal will go on hold. Don't let that sit out there and not get resolved. So the last one is genotype not on file. So that indicates that the parent registration number listed does not have DNA or a DNA sample on file at this time for the calf to be compared to. Therefore, the animal cannot be tested to qualify or exclude as a parent. So when we're taking a look at these results here, you can see on the dam side, genotype not on file, which isn't an issue unless you have a donor dam. Like I said before, all sires and all donor dams need to have DNA on file with Red Angus. For this scenario, it just means that, like I said before, it can't qualify or exclude to the parent because we don't have any DNA to compare it to. Next, we're going to go over some defect results. So basically, this first one you see here says OS free, and that's all the way in the right-hand side. So that means that that animal does not carry the genetic defect listed. A tested free animal is listed with the genetic defect and then the letter F. So for example, OSF. So you may see that sometimes you'll see OS free. The one below that, you can see it says OSC. So that means that that animal is a carrier. Animals that are carriers, OSC, can be registered. However, all future progeny must be tested for the defect as well. We take defects very seriously, and that is why we test them. So if you do have a carrier, like it says, they can be registered. You're just going to have to test progeny out of that animal. Next up, this is kind of two parts. So the very first paragraph kind of goes over the fact that if you did like a GGP test with us, that the data will be applied after the bolt analysis runs. So basically the bolt analysis will pick it up. It'll run for the week. And then after that, it will upload onto the pedigree. Now, sometimes this takes a little bit of time, depending on when we get the results back and when Bolt actually picks it up. If you have any questions about that timeline, feel free to visit our announcements tab through RedsPro or give us a call. Next up is a really big one. So sometimes you'll get sample status, as you can see all the way to the right, finished, failed. So that means that there was nothing the lab could grab from that sample. So there is no data reported on this animal, and that we would need a new sample submitted to us. With that new sample, you would need to fill out the GeneSeek order form as well as payment again and have that new sample. So make sure when you're looking, you see that there, it's been finished or failed, and make sure to be very cognizant of that, because some folks we do see that there is a registration number and they think that we're good to go, and that's not the case, and I don't want anybody to be frustrated with that. Also, another thing to keep in mind is that for twin animals, blood samples are not what you should be testing with. You should either do tissue or hair. Hair cards come with a $5 processing fee on top of your test. So kind of weigh those options, figure out what's best for you. Sometimes if an animal has had multiple samples submitted on it and it's failed multiple times, that might be an inclination that that animal is a twin and something happened when it was born, and we need to get a hair or a tissue sample on it. So that's one thing to kind of keep in mind if you have a sample that continuously fails. So also something to note is if your sample fails, that means it went through 2 rounds of testing and efforts to retrieve data from the sample. So if that happens, you will also get reasons why your sample may fail. And that goes over biological contaminations, chemical contaminations, improper storage, insufficient sample, improper shipping, and that goes over all of that. for your hair cards, blood cards, whole blood, so like blood tubes, your Allflex tags, and even semen. So please take a look at that if your sample has failed. Once again, if your sample has failed, you will need to collect a new sample, submit a new GeneSeek order form for it, and payment. Last but not least, basically these last two statements are just saying that, hey, if you did parentage or defect testing, that will be loaded onto the animal, hopefully by Wednesday. So for example, let's say we got the results back on Monday for your animal and it's OS free. By Wednesday, that should be reflected in your pedigree. Also, another thing is we don't print certificates automatically for animals. So let's say, once again, your animal is OS free. You want that on your certificate that you have at home. So if you wait till Wednesday, you see it's on the pedigree, give us a call and say, hey, can you issue me a new certificate for this animal because it's now showing on the pedigree as OS free. So if you do need that, please go ahead and you can give registrations a call. Their extension is 1, or you give Fallon or myself a call, and our extension is 8 to get those certificates printed out to you. Again, if you have any questions, DNA's extension is 8. Registrations, as a refresher, is 1. You can always email us at dna@redangus.org as well. And also, if you ever need any blood or hair cards, please let us know. This is a service we provide free to you, so make sure you have plenty for this new submission season. I hope you found this video informative, and if you have any questions, please feel free to contact us.